Okay, today we're going to talk about translation. And as before, we're going to focus on prokaryotic translation because much of what we know about translation we learned by studying E. coli. But before we talk about the mechanism, we want to focus on the players in our little movie. And so the first main player is going to be the ribosome. And the ribosome actually consists of two parts, the large subunit and the small subunit. And as you can see, the large subunit has three major components, and we're going to be talking about that as it becomes necessary. The next major player is actually the transfer RNA. Now, the transfer RNA will actually become activated before it is used in translation. And the way that it becomes activated is that it interacts with the appropriate amino acid. And the enzyme that does this reaction is known as aminoacyl transfer RNA synthetase. And what happens is that it uses ATP and it actually allows for the attachment of the amino acid to the appropriate transfer RNA. And this is what comes into the growing ribosomal apparatus. All right? So obviously, in addition to transfer RNAs, we are going to need a large variety of amino acids that will be grown together in our very exciting protein. During the actual act of translation, we are going to need energy. And interestingly enough, with the exception of when the transfer RNA gets activated, the only energy source used in translation is GTP. And just like ATP, GTP uses a series of high energy phosphate bonds. Okay, and this will be the energy source used during translation. Also in translation, we have a series of initiation, elongation, and termination factors. And so we'll be talking about these as it becomes necessary. And then finally, a very important player will be our messenger RNA molecule. And as you can see, it is a large single-stranded molecule that is made through the process of transcription. So all of these will play a very important role in our play. Okay, when E. coli is not translating, both the small and the large subunits of the ribosome are actually together. But when the cell begins to translate, we want these subunits to actually not be together, because the only way that the small subunit can attach to the messenger RNA is if it is separate from the large subunit. So a couple of things need to happen. First of all, initiation factor 1, IF1, will help the large subunit and the small subunit disassociate from one another. And so that's a very important thing. Once that happens, IF3 will attach to the small subunit, and that helps keep the small subunit and the large subunit apart from one another. Now, once this happens, the messenger RNA can now attach to the small subunit. Now, a very interesting thing happens. Messenger RNA of E. coli has a very important sequence at the 5' end. Now, this sequence we've marked here with this little circle, and it's marked SD. Now, SD stands for the Shine-Delgarno sequence. The Shine-Delgarno sequence is actually a small consensus sequence found on the 5' end of E. coli messenger RNAs, in fact, all prokaryotic messenger RNAs. And this actually corresponds to the 16S ribosomal RNA found in the small subunit of the ribosome. They will actually bind to one another, and this helps orient that messenger RNA. And this actually helps the ribosome find the very first it helps orient this, and it helps it find the first start codon. Okay, once we have oriented the messenger RNA, we have the start codon. Now the charge transfer RNA can come in. And when the charge transfer RNA come in, it doesn't come in alone. Several initiation factors come in with it, as well as a GTP molecule comes in. Okay, so here's our transfer RNA, and it's going to line up with it. And notice how when it lines up according to Watson-Crick base pairing rules. So a U will base pair with the A, an A will base pair with the U, and a C will base pair with the G. Now, this is also in accordance with base pairing rules. This is anti-parallel. So this is the 5' end of the messenger RNA. So this must be the 3' end of the transfer RNA, and this must be the 5' end of the transfer RNA. Okay, now the transfer RNA, when it came in, it is carrying the methionine. In bacteria, it's not really methionine. It's actually N-formal methionine, which is slightly altered chemically, just so you know that. Okay, the transfer RNA has an initiation factor that is associated with GTP, initiation factor 2, tightly associated with GTP. Additionally, initiation factor 1 has also come in, and that's going to associate with the small unit of the ribosome. Okay, so this has all come in together. So all of this together now, the small subunit, initiation factors 3 and 1, IF2 plus the GTP, this all together is now known as the 30S initiation complex. All right, so now we have our 30S initiation complex. And so now what's going to happen? The GTP is going to hydrolyze. So this is the energy is going to be released. Our bond has been broken. And what happens now is that all 3 of the initiation factors are going to be released. All 3 will be released. They will float away. And now the large subunit will come in. And when the large subunit comes in, the large subunit will come in, and the first amino acid is actually going to be at the P site in the large subunit. Okay, so now we have the end of initiation, and this is called the 70S initiation complex. So at the 70S initiation complex, we have the F met lined up at the P site, and the ribosome is completely assembled. And now we can talk a little bit more about the E, P, and A sites. So the E stands for exit, and this is where the empty transfer RNAs are going to be leaving the ribosome. The P site stands for peptidyl, and this is where the growing peptide chain will be found, and that's why it's called P for peptidyl. The A stands for aminoacyl, and this is where the amino acid, the single amino acid on our transfer RNA will be coming in. Okay? E for exit, P for peptidyl, A for aminoacyl. We're now ready for the process of elongation. Okay? And we can see at the A site, the next codon is lined up. And so our next charged amino acid is going to come in. And along with that amino acid are going to be 2 elongation factors, EFTS and EF2. And complex with that is going to be a GTP. So all of these are going to come in. And so we have our transfer RNA lining up again with the appropriate base pairing. And then we have our GTP along with the complex of the elongation factors coming in. This will come all in together. And now the GTP will hydrolyze. Once the hydrolysis takes place, the elongation factors will be released. And be regenerated for the next round of elongation. All right, so now in the next step of elongation, a couple of things are going to happen. The first thing that's going to happen is we're going to have a peptide bond form between the two amino acids here. And then we are going to have two more things come in. We're going to have another GTP come in along with elongation factor G. This is going to cause the ribosome to move down in a process known as translocation. And so we have our dipeptide bond has formed, and we see the growing peptide is in the P spot. Our empty transfer RNA is in the E spot, and this will float away. And our A spot is now empty because the ribosome has shipped down in the process known as translocation. So what does that mean? Well, what that means is we are now ready for the next amino acid to come in. And the next amino acid that's coming in, we read our dictionary, and that means the next charged transfer RNA that comes in is glycine. And so glycine comes in along with GTP associated with elongation factor TS and elongation factor TU. So as you can see, hydrolysis of the GTP molecule has occurred and the two elongation factors have been removed. And so now we have a growing peptide in P, the amino acid here. So what do we need? I'll give you a minute to think about it. Did you remember? I hope you did remember that what you need now is another GTP molecule and elongation factor G. Okay, as we can just see, we had enough peptide bond formed. The GTP is hydrolyzed in the presence of elongation factor G, and we've had a translocation of the ribosome. And so now, once again, we have an empty transfer RNA that is floating away, and we have our growing peptidyl chain on our P spot, and that leaves an empty A spot waiting for the next transfer RNA to come in. And that, of course, would be corresponding to our ACC codon, and that would be this charged transfer RNA. That would come in here along with the GTP complex with our two elongation factors. And in the same way, this GTP is going to hydrolyze. And when it hydrolyzes, the two elongation factors are going to disassociate from it as well as the GTP. This disassociation is now going to mean that the GTP will come in along with elongation factor G. And this is going to signal that the next peptide bond is going to form. GTP is going to hydrolyze again, and now we are going to have the movement of the ribosome down again. This will shift the growing peptidyl chain to the P, and we are going to have an empty A again. Now once again, on the E site, the exit site, we have an empty transfer RNA which is going to float away and be recharged again by amino acyl transfer RNA synthetase. And I could continue doing this for as long as I wanted to, but I would get bored and so would you. So what I want you to do is to actually look at the rest of our messenger RNA and make sure that you can do both the translation And make sure that you also know what the anticodons are. Okay, so we've completed the elongation phase of translation, and now we're moving into the termination phase. And there are 3 termination codons. There are no tRNAs that bind to the termination codons. Instead, there are release factors. And these release factors will actually bind at these codons. And when the release factors bind, several things happen. The release factor will bind at the codon. And depending upon the codon, either release factor 1 or release factor 2 will bind. When that happens, then release factor 3 will bind to the large subunit. Release factor 3 is associated with, once again, GTP. When this happens, GTP is hydrolyzed, and this releases some power. And this hydrolyzing causes several things to happen all at once. It causes several other factors to kick into gear. And one of the things that happens then is this bond is cleaved, and the protein that you have made is now released to go about and do whatever it is it's supposed to do. So it will go ahead and be on its merry way. The transfer RNA is released. The ribosomes disassociate from one another, and the messenger RNA floats away. The release factors disassociate as well, and everything falls apart. So there you have it, translation in prokaryotes. One thing to remember about prokaryotic translation is that messenger RNAs tend to be polycistronic. This means that there's more than one gene located on the same messenger RNA, so that there tends to be a start codon, a stop codon, and then immediately following that stop codon, another Schindel-Garneau sequence, another start codon, and another stop codon. And there can be as many as 5 or more on the same messenger RNA. This is not true for most eukaryotes. Eukaryotic translation is actually, the mechanism is very similar. What is different is the associated initiation factors, elongation factors, and release factors. Additionally, there isn't a Schindel-Garneau sequence, but there is a similar sequence called the Kozak sequence, and that helps orient the message so that it can help find that first initiation sequence. I hope you've enjoyed the film, and look for the next exciting film. Bye-bye.